Nice to meet you all. Like said, my name is Krista Rantanen, and I'm the Director of Scientific Applications at Ruskinn, based in Finland, so that's where I came. Nice, comfy, minus 30 Celsius going on this week, so I'm kind of appreciating the not minus 30, so that's pretty cool. So my background, I'm an academic for some time. And what I did was that I studied hypoxia, and specifically I studied proline hydroxylase enzymes in the setting of kidney cancer, or the basic molecular biology. And then some years ago, well, in fact, when I was working in the lab, I had the Ruskinn workstation, the kind that you're going to see today, with the glove box. And so that's what I had in my lab, and that's what I worked with for 15 years or so. But I love the topic so much that I actually decided to join Ruskinn. So thanks for hiring me. And ever since, this whole thing has been my life. Today, though, I'm not going to talk so much about biology or science, other than just the basics, which Giovanni already touched upon. A little bit of that is going to happen. However, I just wanted to introduce to you something from our really neat product portfolio that enables exactly what Gio was talking about, how to do physiological cell culture. Now, I could talk about the workstations, but then I chose to introduce something completely different, something that you may not have heard of and may probably not have used. And this thing is called Oxygenie. Now, before I actually go to Oxygenie, let's take a look a little bit what you're going to hear. There is terminology, a little bit, just Giovanni went through that, but I just want to highlight something that there won't be any misunderstandings while I'm talking. Just a teeny tiny bit of scientific background, and then all about Oxygenie, a thing that's called Oxygenie. So, like you now know, we have this concept called normoxia. So when you read scientific papers, this is what you hear, right, or read. And if you think about it, it kind of refers to something being normal oxia, say. But biologically, that means, again, it's the 21% of oxygen in the ambient air, or perhaps 19.8 in the incubator humidity setting. And again, like we saw in Giovanni's presentation, that's not what happens in the body, okay? Another term that you often hear is hypoxia, but in a biological setting that refers to something being a little bit wrong. It's a malignant condition which refers to oxygen levels being around 1% or less. Okay, so two very ends of the story, normoxia and hypoxia. But in between we have something else. We have physoxia, so physiological oxygen, and that's what we're here for. Okay, and that would be, of course, like I said, depending on tissues and organs, varies somewhere between 13 and 3. Not really important here what the actual level in each organ is. As you can see here, they vary. What is important for this talk, however, is to understand that it's never, ever 21% of oxygen. Nowhere, the cells that you study, so in a biological setting, they never see 21% of oxygen. And sometimes when I say this, people go like, well, that's a bold statement. But the fact that it's not a bold statement, it's just a biological fact. And so when we do research, and we do it in normoxia usually because it's very handy, it's easy to do, we might want to keep in mind that that's perhaps not the in vivo situation. Okay, so there's that. What happens if we then, however, decide to use the incubator, which has about 19.8% of oxygen? Well, loads of things happen, and unfortunately for us scientists, the loads of things that happen are usually bad things, because at the end of the day, when cells and enzymes work, they're range of operation, say enzymes, it's within really narrow range of oxygen. They cannot handle high oxygen. So this is hyperoxia, what we do, and I refer, we, the scientists that do the work. So what we actually do is that if we grow cells at 21, we introduce, for instance, reactive oxygen species bursts, okay? So obviously that's enough to kill or modify the function of the cell or the enzymes or proteins or anything. So that's a very bad situation. Hypoxia, well, obviously if you have too little of oxygen, oxygen is the energy source, and if you don't have oxygen, then the cells have very little of energy. However, in the setting of, say, cancer, that's not such a big problem. Cancer cells have this really funny tendency to just flourish under low oxygen, okay? This is because tumors, when they grow, they will so quickly outgrow the supply of oxygen that they will form adherent regions of low oxygen, and they will adapt and still grow. So even though for a general setting, normal cells will experience lack of energy in cancer setting, there will be upregulation of glycolysis and VEGF and angiogenesis occurring, which of course helps, for instance, the cancer to grow. But anyway, so the point of this slide is to show that, this is something that we hardly ever do by accident, but this is what we do in the lab, and it's always, we don't really think about it, that we put them in the incubator, that it's wrong, or it causes harm to the cells, but just keep that in mind. Because at all costs, and I'm going to, in the later slides, you will constantly see two phrases, and it's compromised results or untranslatable results, that they're not reproducible, okay? So you know, as scientists, you know how important it is to have reproducible results. No journal is going to publish anything if it can't be reproduced somewhere independently from your lab, correct? And where does the variability for results come? Well, it begins when you start using, when you have your material, when you have your cells. And if you start to treat them in a way that has loads of variables, then your end result will inevitably be possibly untranslatable, and you can't reproduce that data. So just as an example, we have cells. We got them from ATCC, but maybe they came from a mouse. Okay, so we have cells. And the cells are quite happy because they are in their environment where they naturally reside. Now, in this mouse setting, in here, the oxygen concentration, like I showed in tissues, is quite low, okay? So here it goes. But then when we start doing our experimentation, we transfer the cells in a BSD. We give them media and supplements, and that's one thing, but we also subject them to significantly higher oxygen concentration. Now it's the 21% of oxygen. The cells are quite unhappy. Then we transfer the cells into that CO2 incubator, and if it's a regular CO2 incubator, it'll be, again, high oxygen. If you're lucky, you have a tri-gas incubator where there's low oxygen, which is way better. But the cells are unhappy nevertheless because they have experienced this part of the journey, which has been stressful to begin with. Okay, so we have them here, and then when we do the final examination or final experimentation or whatever your endpoint assay is, it's usually done in very high oxygen at 21% of oxygen. So the cells are unhappy. And what we see here is this fluctuation, and it varies and it produces very inconsistent results, because this kind of thing is virtually impossible to mimic anywhere, correct? So this is what we want to take away from the experimentation. So how do we do that? Well, we have those workstations that you're going to see, but we also have something else, and this is what I want to talk briefly about, and we're going to go to the lab and we're going to have a hands-on session with this product. So this is not going to be a technical presentation, I'm just going to walk you through what Oxygenie is. It's a benchtop physiological system. So when you have the big workstations, you might as well have a small one as well. And here it is in the trunk of my car. And here is an Oxygenie benchtop physiological system carried by Dr. Carol Hells, who's our science director in the US. So this is Oxygenie. Essentially, what Oxygenie wants to do and is designed to do is that you have a very nice scientific workstation. This is an incubator inside. Okay, the cells are kept in a nice warm temperature in low oxygen. But this is a big chunk of a thing. So Oxygenie, as you can see, is a much smaller thing, but essentially it does what this piece of equipment does. It just is made very much smaller. And how it works, so here we have these colorful little things that we call one wells. And this is where your cells are seated. So these are the size of a 12 well plate, the diameter. So this is where you plate your cells, just like you would on a regular cell culture plastic setup. It sits on a heat plate, so they are very comfortable. They are kept in a warm environment. And then, and this is the thing. We have two gas cylinders, and they are filled with premixed gas. And we have tubing coming through here to this system. As you can see, there are little tubes here. This is called the flow divider. And the tubing now divides the flow of gas into these small one-well structures. Now, what's in here could be 5% of oxygen, 1% of oxygen. So essentially, you create that atmosphere in here. Okay. What Oxygenie is used for, differently from a workstation, is that this is for cell culture studies, or you can use other material for growth. But you can also do imaging. You can do high-resolution imaging with this system. So not only observing cells, but you can actually, in high resolution, see live cells behave, how they behave under low oxygen. You can use this to transport material from one place to another, which is usually and many times very, very beneficial when you want to avoid that that high oxygen gap when you take your samples from one place to another. So where we use Oxygenie, and I'm going to talk about these more when we actually see the product, but you can use it in imaging, irradiation. This is pretty cool. I'm going to talk about that a little bit later. You can use this in conjunction with the workstation, but also very beneficial if you don't have access to a full-blown workstation system. For transportation, but then personal favorites are plant sciences and small animal studies as well. And just going through how this works. So imaging under hypoxia is really difficult. You cannot, if you have a workstation, then you would possibly have, you might think about putting a microscope inside your workstation. But now imagine that's a little bit complicated and perhaps a little bit pricey, but you can do that, it's possible. Another solution, if you just choose not to do any imaging with yourselves, but you lose quite a bit of data if you don't do any imaging or don't give yourself the possibility to do so. Or, in fact, this third option is something that people usually do. They have their cells in the workstation and then they take them out for just a moment and quickly go and image. But then that's impossible. Like I said, enzymes are really sensitive, but they are also very quick. So you actually do not have any time you have minutes to work before enzymes start to be harmed by high oxygen. So that doesn't really work. There's going to be reoxygenation damage if you do that. So what happens with oxygen is that you actually take this whole system, the cells in hypoxic conditions, in low oxygen, next to your microscope, say it's a high-end confocal, okay? And you can actually detach this plate and put it on the microscope stage. It's a K-frame, so you can actually have this this heated stage on the microscope stage and have live cell imaging under hypoxia. So that's one way of using it. Then we have the irradiation, and this is actually what it was designed for. Like I said, tumors are always hypoxic. However, if you look for studies that show how cells behave in combination or respond to combination of hypoxia and irradiation, you can't find it. Excuse me. And this is because it's technically quite challenging to have a workstation where you have irradiation. So what happens here is that you take these, you again take the whole box next to your irradiation machine. The cells are in hypoxic conditions. You can detach these one wells, put them in the irradiation machine, zap them, and put them back, and the cells never knew what happened. Excuse me, you can tell that it has been a long time since I've spoken in front of people. Teams doesn't make you cough, by the way. I don't know how that is possible.
You have been vaccinated, right?
3 times. And tested it like zillions of times even before coming here. I'm so negative, it's like not funny. I'm just so negative. Anyhow, so irradiation is my personal favorite. Again, like I said, you can use this if you have a workstation. So you just take these one wells, this is the structure, and I will show you them later. You just throw your cells in these ones in the workstation. And then if you want to do imaging, if you want to do irradiation, you just put them in this transportation device as it now stands and take them to your facility where you want to go. So this really adds flexibility and options for workstation use. If you don't have a workstation, oxygen is really good because it enables doing proof of principle studies, for instance. If you want to prove to your grant, write in your grant application that this actually makes a difference, but you need proof of principle studies to show that. And this is the kind of a solution that you can use for that one. Of course, less pricey than to obtain full-on workstation for sure. And you can do the imaging and all the other things that was mentioned. So it does have a very good function without a workstation as well. Transportation. So oftentimes, imagine that you want to get samples from operating theater, but then we face that nasty reoxygenation damage as soon as the cells are collected. So you can actually take tissues, bits of tissue, and put them in here in low oxygen directly. Or again, if you need to go to an imaging facility with your cells, usually you don't have a high-end microscope in your lab. Good for you if you do, but usually people don't. And it's an imaging facility. So you can use this for transportation, for keeping your cells unharmed through the process. So it kind of bridges the gap between 2 low oxygen locations and avoids that reoxygenation. For plant sciences, and this is one of my personal favorites because as you possibly know, it has been only recent years when we have realized that it's quite conserved throughout species how cells respond to oxygen. So the mechanism is quite similar actually in plant cells as it's in mammalian cells, for instance. And we know this now. Why it's important to understand how plants behave in hypoxia, you wouldn't think about it, but in this world of climate change and everything, we know that flooding incidents are growing. And they usually occur in regions where it is a huge socio-economic disaster, because those crops that flood feed a whole lot of people. And it's usually based on that one type of plant. So to understand how roots and seeds survive or don't survive hypoxic conditions underwater, there is no oxygen underwater, as you know, hard to breathe. It would give invaluable information on perhaps making these crops more tolerable in flooding and especially when the water goes away, which is again that re-oxygenation damage. So you can actually put roots and seeds in here and mimic a flooding situation. So that's quite neat. And last but not least, a lot of studies is going back to using small model organisms, and this is of course because less paperwork, population doubling time is quicker, or those sorts of things, and plus they are all feed-through. So really good for developmental biology and genetics, and you know this thing. But what you maybe didn't think about that is that you can actually place in these one-well structures, you can put worms, you can put flies, or fish embryo in here, and in a controlled manner deliver low oxygen surroundings and study the responses. So that's pretty neat. That's a different thing. What I've just told you and what I will keep on telling you when we go into the lab is that we have nice key features with Oxygene, the new product. So live cell imaging in high resolution of cells in low oxygen. Really hard to do as it is. Irradiation of live cells followed by possibly high resolution imaging. Hasn't been possible before. Move cell lines around, move samples around in physiologically relevant conditions in a very precise manner. So there is no evaporation, which means no changes in pH or other factors. And we have the 3 Ps. So it's portable. You can try. It's 8 kilos, so you can try and lift it. Physiological and extremely precise. So all these things we're going to go through when we go into the lab, and you can actually play around. It's a really neat little tool. You can open and recompose the one wells and try it out in any way you like. So this is where I stop, and I think that's where I stop. And yeah, we have questions later. We have questions later. Thank you so much. Thank you.